Welcome to the deep dive. I want you to picture something with me to start things off today. Just imagine you are standing in a glowing, three-dimensional holographic forest.
Oh, that's a pretty intense visual.
Right. It's vast and dark, but as you look closer at the trees around you, the trunks start to morph, the branches begin to shift and pulse with light. And you realize pretty quickly that you aren't looking at a forest of wood and leaves at all. You are standing inside a microscopic forest of human brain cells.
It is a beautiful, almost, intimidating image to hold in your mind. And honestly, it is the absolute perfect way to visualize the sheer scale and complexity of what we are going to explore today.
Exactly. Because today, our mission is to answer a question that sounds incredibly simple on the surface, but has historically been, well, almost impossible to prove.
Yeah, it really has been.
And here it is. Is the human brain just a magically scaled-up mouse brain? Think about it. If you take a mouse brain and just pump it full of air until it's the size of a cantaloupe, is the microscopic hardware exactly the same?
Right. Or is our actual cellular wiring fundamentally architecturally different?
Exactly. And to finally answer that question, we are pulling from a truly groundbreaking 2026 paper. It was published in Nature Neuroscience.
Yeah, the title is a bit of a mouthful. It's called A Framework for Comparative Analysis of Human and Mouse Cortical Neuron Dendrites in Corresponding Brain Regions.
Yeah, it's a very dense title.
It is, but it is a monumental piece of research. I mean, it finally gives us the mathematical and biological tools to compare these two biological machines side by side.
Right, so let's establish the core mechanism here so everyone has the blueprint. We are talking about something called dendrites.
Right, the branches.
Yeah, so if a single neuron is one of those trees in our glowing holographic forest, the cell body, what biologists call the soma, is the trunk. The dendrites then are the branches. They reach out and receive all the chemical whispers and electrical signals from the rest of the forest.
And in this deep dive, we are focusing strictly on their morphology, meaning the physical shape, the architecture, the exact branching patterns of these dendrites.
That's a crucial distinction. We aren't looking at the overall size or weight of the brain.
No, not at all.
Or even the chemical neurotransmitters. We are looking at the specific microscopic geometry of the brain's receiving antennas.
Which, raises an obvious question for anyone listening. Why hasn't this been done before?
Right. I mean, we've been dissecting and studying brains for centuries. Why is a side-by-side comparison of human and mouse brain branches a 2026 breakthrough?
Well, it really comes down to three massive historical roadblocks that scientists just couldn't easily bypass.
Okay, lay them out for us.
First, there is a sheer scarcity of human three-dimensional reconstructions. Getting living, functional human brain tissue to map in highly detailed 3D is, well, it's incredibly difficult for obvious ethical and medical reasons.
Yeah, you can't exactly just ask someone to donate a slice of their healthy brain for a map.
Exactly. And second, there is the mapping problem. A mouse brain is completely smooth. A human brain is incredibly wrinkled, full of those deep valleys and ridges we call sulci and gyri.
Wait, I'm trying to visualize this. A mouse brain is smooth like a jelly bean, but our brains are wrinkly like walnuts.
That's a great way to picture it, yeah.
So if I'm a researcher, I can't just stretch the jelly bean over the walnut and expect the regions to match up, right? How do we even know we are comparing the same functional real estate?
Exactly. You can't just overlay a smooth map onto a wrinkly one and declare that a spot on the left side of one equals the left side of the other. The anatomy just doesn't translate perfectly.
Right.
And then there's the third roadblock, which is the scale problem. Human cells are just inherently bigger.
So if you compare a giant human cell to a tiny mouse cell, of course they look different to the naked eye.
Right. How do you mathematically level the playing field so you are only comparing the design, not just the raw size?
It's like trying to compare the architecture of a sprawling suburban mansion to a tiny studio apartment, but you're only allowed to look at the plumbing.
Yes.
You have to find a way to standardize it, or the comparison is completely useless.
Exactly.
Okay, let's unpack this with a quick recap before we move on. Bullet 1. We're looking at a 2026 Nature Neuroscience study comparing single neuron dendrites across species.
Right. And bullet 2. Our sole focus is morphology, the physical structural shape of the brain's microscopic branches.
Spot on. And bullet 3. The challenge has always been tissue scarcity, aligning a jelly bean to a walnut and equalizing the sheer difference in size.
That perfectly frames the problem.
Which brings me to a question I want you, the listener, to keep in mind as we dive into the solutions. Think about what happens in your own head when you learn a new skill, like learning to play the piano or trying to speak Spanish. It's easy to assume your brain is just growing longer, sprawling branches to reach new areas and make new connections. But what if it's doing the exact opposite? What if it's just packing tighter? Keep that question running in the background.
Because that tension sprawling out versus packing tightly is exactly what this study manages to resolve.
Yeah.
But to get there, we really need to look at how they gather this data, because you can't compare the individual trees until you have accurately mapped both forests.
The scale of the data gathering here is just mind-blowing. Let's start with how they mapped the mice.
Okay, yeah.
They mapped 100 whole mouse brains that yielded over 16,000 individual cortical neurons. And to pull this off, the source mentions they used a technique called fMOST imaging. What exactly is that doing?
So fMOST stands for fluorescence micro-optical sectioning tomography, which is a mouthful.
Yeah, that is a lot of syllables. What does it actually mean in practice?
Basically, instead of just looking at the outside of the brain, this technology allows researchers to image sparsely labeled neurons across entire mouse brain at incredibly high resolution.
Oh, wow.
Yeah, and basically, this is crowdsourced human curation. Human experts step in, look at where the AI got lost in the woods, and manually untangle the branches. It is a perfect human-AI tag team.
Okay, so that's the mouse forest mapped.
Yeah.
But the human data is where this gets really wild. They gathered data from 23 human patients who are undergoing brain surgery.
Right, mostly for tumor removals.
Now, you obviously can't take healthy brain tissue from someone just for an experiment. So the researchers used the clinically normal cortical tissues from the margins of these surgeries, the tissue they had to remove anyway to get to the tumor.
It is an incredible ethical and scientific workaround. And from those 23 patients, they managed to reconstruct over 2,300 neurons from the frontal, temporal, and parietal lobes.
And for the human tissue, they couldn't use that same whole brain optical slicing method, right? Because they didn't have whole brain.
Exactly.
They used something called AC tomography, right? Adaptive cell tomography. From my understanding of the paper, they took these incredibly thick slices of living human brain ex vivo. We're talking 200 to 600 micrometers thick.
Which in the cellular world is just huge.
Right. And to map the trees within that thick slice, they actually injected a fluorescent dye called lucifer yellow directly into the cells.
I love that visual. It's literally like injecting glowing neon sap into the trunk of a single tree and then just watching it flow outward.
Lighting up every single twig and leaf in the darkness.
Exactly, yeah. And then they capture it with advanced two-photon imaging. It's a brilliant technique.
It is.
But here is the crux of the problem. You now have a highly detailed, beautifully rendered map of 16,000 mouse neurons and 2,000 human neurons. How do you match them up? We are back to your jelly bean and walnut problem.
Right. If I have a neuron from a specific ridge on the human frontal lobe, how do I find its exact twin in the mouse brain?
Well, they used a highly sophisticated multi-step matching process. First, they mapped each brain to its own species canonical template.
Okay, break that down for us.
For the mouse, it's a standard template called CCFV3. For humans, it's a nonlinear template called ICBM 2009C.
Okay, those are just alphabet soup to me right now.
Fair enough. Think of this like taking a unique, lumpy clay sculpture of a brain and carefully mathematically warping it to fit perfectly inside a standardized master mold without losing any of its internal coordinates.
Okay, so now they have standardized the mice to a master mouse mold and the humans to a master human mold.
Right.
But how do you cross the bridge between the two species? How do you prove that region A in the mouse is functionally region A in the human?
This is where the paper really shines. They use transcriptomics.
Gene expression. This is the good stuff. Explain exactly how this bridges the gap.
Well, even if the macroanatomy, the jelly bean and the walnut, looks completely different to our eyes, the underlying genetic code driving the cells in a specific region can be nearly identical.
Really?
Yeah. A transcriptomic profile is basically the cellular recipe. It tells us exactly what proteins and molecules that specific population of cells is manufacturing.
This is like a biological barcode.
Exactly, a barcode. The researchers looked at the transcriptomic cell composition profiles across both brains. If a specific region in the human frontal lobe has a genetic barcode that statistically matches a region in the mouse brain, they declared them corresponding pairs.
So they stopped guessing based on physical landmarks and used biology's own source code to align the regions perfectly.
Exactly. It's incredibly elegant.
But even with the forests mapped and the regions perfectly paired up through their genetics, we still have that third roadblock.
Right, the scale problem.
Yeah, a human cell is just bigger. If you compare a big tree to a small tree, the branches will look different simply by virtue of being larger.
And that leads to a critical mathematical mechanism in the study, which is size normalization.
Okay.
Before they could compare anything, they measured the actual cell bodies, the somas or the trunks of all these neurons, and they found a very consistent ratio.
I love it.
Human neuron somas are exactly 1.68 times larger in diameter than mouse somas.
So to make it a fair fight, they instituted what I like to call the 1.68 rule. I really need a visual to grasp this, so picture this. You have your glowing holographic mouse tree in front of you.
Mm-hmm.
You take a digital magnifying glass and you mathematically blow up the mouse tree's trunk by 1.68 times. You scale all the branches up with it proportionally until the mouse trunk is the exact same size as the human trunk.
Exactly.
And now raw size is completely removed from the equation. Now we are only looking at the architecture itself.
It's an absolutely crucial step. I mean, without that 1.68 scaling rule, you might look at a human dendrite and say, wow, the branches are so much longer, human architecture is so advanced.
Really, it's just a bigger cell.
Right. By scaling the mouse neuron up to match, we eliminate the illusion of volume and isolate the reality of structure.
So let's unpack this and get our bearings with a quick recap on how they built this foundation.
Yeah.
Bullet 1. They gathered massive data sets using fMOS for the mice and AC tomography dye injections for the humans.
Yep. And bullet 2, they didn't just guess the anatomy. They matched these regions perfectly by reading their genetic barcodes.
Right. And bullet 3, they used that 1.68 rule to scale up the mouse cells, mathematically guaranteeing a perfectly fair size normalized fight.
That is the foundation. Every variable is controlled for.
Which brings me to a question for you to ponder. If you take size completely out of the equation, if a mouse neuron and a human neuron take up the exact same relative footprint, where would you guess the human brain puts its extra evolutionary effort?
Yeah. If we aren't just building wider, longer branches, what are we building?
Exactly.
The answer to that question completely rewrites how we think about cognitive complexity. Because once the map was set and the sizes were equalized, they finally looked at the branches.
And what did they find?
When they looked at the branching frequency, and specifically the intervals between those branches, the researchers hit a massive breakthrough.
Oh, wow.
Yeah. Even when normalized for size, human neurons have a vastly higher branching frequency and remarkably shorter branching intervals.
Let's break down how that actually works, because the paper talks a lot about branch order, or BO.
Right. Think of branch order as a way to map the generations of a tree. BO1 is the first primary branch coming right off the trunk. BO2 is when that first branch splits into two new ones. BO3 is the next split, and so on.
Makes sense.
The researchers found that in humans, those lower order branches, the basal ones right near the cell body, BO1 and BO2, are incredibly short.
Meaning they don't travel very far before they split again. Humans use these relatively short branches to bifurcate, to split into new paths almost immediately.
Exactly. They bifurcate rapidly and repeatedly.
This is where my absolute favorite analogy of the entire deep dive comes in. Think of the mouse neuron like a wild, sprawling oak tree.
Oh, I like that.
Yeah, it has these long-reaching, linear branches that just stretch out lazily into the space around it, going a long distance before deciding to split.
Right.
But the human neuron, it's like a meticulously pruned bonsai tree. It is hyper-compact. It turns and splits and bifurcates constantly, packing an astonishing amount of surface area and complexity into a very tiny, confined physical space.
It is the ultimate expression of spatial efficiency in biology. The human brain isn't just sending a long wire from point A to point B to make a connection.
No.
It is building a hyper-dense, complex mesh of receiving antennas right at the source. It is prioritizing local integration over sprawling reach.
And this spatial organization leads to another huge discovery in the paper regarding what they call lobe separability.
Yes, this is fascinating.
I had a real moment of curiosity reading this part of the sources. Does a neuron in the front of my brain, say in the frontal lobe, look fundamentally different than a neuron in the back of my brain, in the parietal lobe?
The answer is yes, they do look different depending on where they live. But the really fascinating part is the mechanism of how they differ.
Right, because it's different in mice and humans.
It is completely divergent between humans and mice. The paper highlights this beautifully. When the algorithms try to mathematically separate mouse neurons by their lobes sorting them into frontal versus parietal versus temporal, the mice neurons differentiate based on what researchers call branching topology.
Topology, meaning how deep the branches go.
Exactly.
The sequence of the splits, like counting how many forks in the road there are from the trunk to the final leaf.
Topological depth, yes. Mice organize their brain regions by how deep the oak tree's roots and branches go.
But human neurons.
They differentiate based on spatial organization. It's not just about the linear sequence of splits, it's about how those branches physically pack into three-dimensional space.
Wow.
Yeah. When the algorithms sort human lobes, they rely on metrics like 3D density and center shift.
So if I'm a researcher looking at a human brain cell, I can tell what lobe it belongs to not by counting the number of splits, but by measuring how densely packed and physically shaped the bonsai tree is?
Precisely. If we connect this to the bigger picture, it implies a massive evolutionary leap. Human neurons are manipulating 3D space in a way mouse neurons simply aren't. We are sculpting our hardware to fit the space.
And nowhere is this incredible spatial density more apparent than when the researchers zoomed in on the frontal lobe.
Oh, this is my favorite part of the data.
Let's do a deep dive within the deep dive on the frontal lobe. The researchers matched a highly specific pair using those genetic barcodes we talked about.
Right, the transcriptomics.
Yeah. They matched the human superior frontal gyrus, or SFG, against its exact genetic counterpart in the mouse, which is the secondary motor area, or MOs, and the results were just staggering.
The human SFG showed the absolute highest discrepancy in branch abundance of any matched pair in the entire study.
And we have to ask why. Why is that specific spot so wildly different?
Well, the superior frontal gyrus in humans is ground zero for advanced executive functions.
Right. Think about your day-to-day. When you hold a thought in your working memory, when you plan out a complex series of physical movements, when you process a high-level cognitive problem that is your SMG at work.
Exactly. And to pull off that level of thought, you need immense signal integration. You need millions of different cellular whispers combining into a single cohesive command in a fraction of a second.
So the human brain solved the problem of executive function, not by just making a bigger, longer oak tree to reach more areas, but by engineering the richest, most intricately split, hyperdense bonsai tree structure in the entire brain.
It's amazing.
The branches in the human SFG are just exploding with bifurcations compared to the mouse.
It really proves that our cognitive architecture is completely bespoke. It is highly customized for the specific computational needs of human intelligence.
It's not just a bigger engine, it's an entirely redesigned circuit board.
Perfectly said.
Okay, let's quickly unpack our steps through these findings because this is dense material. Bullet 1. We learned that human neurons don't just grow longer, they bifurcate much more frequently with shorter intervals, creating that bonsai tree effect.
Right. And what's fascinating here is bullet 2. When distinguishing between different brain lobes, human neurons differ based on their 3D spatial packing and density, whereas mice differ based just on topological depth.
Exactly. And finally, bullet 3. Our human frontal lobe, specifically the SFG, shows the most extreme architectural upgrade of all, packing in unprecedented complexity to support our advanced executive functions.
That covers the key takeaways perfectly.
Which brings me to a question for you to reflect on right now. Think about your own workspace or even your computer desktop.
Oh, this is a good one.
Are you organized like a mouse neuron, sprawling linear, taking up a ton of surface area to get things done? Or are you organized like a human neuron, hyperdense, spatially optimized, packing maximum utility and complexity into a very small footprint?
It's a great way to visualize the difference in efficiency.
So we've walked all the way through this microscopic holographic forest. We've scaled up the trees with the 1.68 rule, mapped the genetics, and looked closely at the densely packed branches. We are in the outro now, the big synthesis.
Yeah, the grants. What?
Exactly. What does this all mean for the person listening to this right now? How does this incredibly microscopic architecture connect back to our daily waking experience of consciousness, of learning a new language, of remembering a friend's face?
The grand so what's of this entire 2026 Nature Neuroscience study is this. It fundamentally disproves a very old assumption that human cognitive superiority is simply a matter of volume.
Right.
Yes, our brains are physically larger than a mouse's, but our actual microscopic hardware evolved a completely divergent organizational principle.
Yeah.
We didn't become smarter just by getting bigger. We became smarter by becoming master space savers.
We became the ultimate biological engineers of efficiency.
Exactly. By creating these hyper-compact, incredibly dense local dendritic architectures, these intricate bonsai trees, our brains allow for massively complex signal integration. We can compute more data, cross-reference more memories, and generate infinitely more complex thoughts with without requiring a head the size of a beach ball to fit all the wiring. We perfectly optimized the space we had.
It's the difference between building a sprawling suburban city of single-story houses that stretches for miles and building a dense, highly connected metropolis of skyscrapers in a tiny footprint like Manhattan.
That's a perfect analogy.
The physical footprint might be constrained, but the activity, the speed, and the connection inside is exponentially higher.
And that extreme spatial efficiency is the physical foundation that gives rise to human cognition.
Which brings me to a final provocative thought for you, the listener, to mull over as we end this deep dive.
Okay.
We've just learned that human evolution solved the problem of higher intelligence, not just by building a bigger forest, but by engineering a radically denser, more intricately connected bonsai tree.
Right.
But physics is physics. Our skulls can't get much bigger for biological birth reasons, and at the microscopic level, biological cells can only pack so tightly.
Right. There is a very real biological ceiling here. If you pack cells any tighter or make them bifurcate any more densely, they start running into fundamental limits of physics. They demand too much energy, they generate too much heat for the brain to dissipate, or they literally begin to crowd each other out of the physical space. Biology has its absolute limits.
Exactly. So here's the question I want to leave you with. Have we reached the biological hardware limit of this compactness?
Oh, wow.
If the human brain is already the ultimate space-saving bonsai tree, flawlessly optimized for the space it has, well, what does the next evolutionary leap in intelligence even look like? Are we maxed out on biological density?
That is a staggering thought.
And if we are, does that mean the next major leap in cognitive architecture has to be synthetic?
This study really pushes us to consider exactly those kinds of profound questions about the ultimate limits of biology.
Keep questioning the world around you and never stop looking closer at the forest. Thank you for joining us on this deep dive into the architecture of the mind. We'll see you next time.